Hello students, a very good morning to all of you, and we have reached the end of the module 11, where we are discussing about the non-metallic elements and their biological roles. And today we will be talking about chlorine as well as iodine. It is not chlorine and iodine, but it would be chloride ion and the iodide ion. So, we will talk about these two elements in biology, whether we need some ion channels for that purpose or not, and why we require some exclusively the chlorine transport or the chloride ion transport for a particular point. And that way we can also try to know that whether Cl minus and I minus, the inorganic anions, the way we know them, are essential for our life processes also. Then how we can correlate this, whether your chloride or your iodide can be correlated to some essential hormone synthesis. And that way also we will be able to know or learn about the iodination and deiodination reactions, which are very important organic chemistry reactions. So, if we have chlorine as well as the chloride ion, because we know that it will be converting to sodium chloride like salt, so which is an essential element and sometimes we call as a very beautiful or very useful nutrient for us for our metabolism, because everywhere we need the presence of chloride ion in our system. So, how much you have? When you first time learned the chemistry practical in your school, you know that your chloride sample is given in the form of sodium chloride, and you have been asked to identify the salt as sodium chloride, not as sodium chloride, but only the anionic part, because it is given that okay, you have given some chloride salt. It can be sodium chloride, it can be potassium chloride, or it can be calcium chloride. So, we have identified that chloride ion by adding silver nitrate solution, and we know that we get the silver chloride precipitation. So, the same understanding or same knowledge we can use also if your sample is different, not we have prepared it for you by dissolving sodium chloride, or not you can have the biological fluid samples. But how much you have, that means the quantitative determination, not only the presence of chloride ion in this biological system or the fluid, but you should know how much is there, which is important, because the concentration plays a very important and vital role for our life processes. So, in the sea water we all know that the potassium chloride, in chili saltpeter we know the sources and all this. So, the geological point of view or the biogeological point of view, where from we are getting all these things. So, highly concentrated sea water can have the chloride concentration up to 0.55 millimolar. Is easy to remember also. We know sometimes we ask what is the concentration of water in water. That figure is also 55 molar concentration, and it is 0.55 millimolar. So, it is 0.55 millimolar concentration what we can have in sea water. So, if some standard reference you have, that means this much chloride ion is present in sea water, and how much they are in our drinking water, we talk in terms of the ppm level of the chloride ion as well as the other ions like sodium and other ions. So, we can have the cell, and outside cell you have some fluid, and inside cell we have some fluid, and we have the barrier, the lipid barrier or the phospholipid barrier we know, which is encasing your cell. So, what should be the outside environment concentration, which is nothing but is 103 milli equivalent per liter. I am not talking in terms of your millimolar concentration, because I am giving you some confusion. So, you should be confused little bit such that you understand nicely how much these equivalences are: millimolar concentration, milli equivalents per liter, as well as the ppm concentration what we drink as your water. So, it is 103, the figure is compared to that of 142 for sodium ion and 27 for bicarbonate ion. So, all these will come and compete with the chloride concentration. Then compared to your outside concentration to the inside concentration, which is very low. So, which is only 2 milli equivalent per liter compared to your bicarbonate concentration, which is high. So, we should know about the balancing of all these concentrations, how much sodium concentration we have in the sodium ion concentration in the extracellular fluid, and how much inside. Similarly, the potassium concentration, we know the relationship when we have learned about the corresponding ion channels of sodium as well as the potassium in biological inorganic chemistry. So, there we have seen that you can have some reverse thing, that means you can have one particular point your sodium is concentrated and another part the potassium ion is concentrated. So, that gives all these informations and all these activities and the functions. So, we can have the typical significance and from physiological origin what you can have, what basically the corresponding chloride ion can do. We know the person suffering from hypertension, we always prescribe that you do not take much of your sodium chloride. So, your sodium intake as well as the sodium chloride intake should be reduced. So, what is controlling that osmotic pressure? You have to know that, you little bit you try to understand it, that what is being balanced, the osmotic pressure due to sodium ion or osmotic pressure due to chloride ion. So, here we have the answer that okay, the osmotic pressure is being controlled by the chloride ion also. What about the sodium ion? So, already we have learnt it, but we can fix these questions or we can give these questions in that particular form, that you compare the role of Na plus and role of Cl minus in controlling the osmotic pressure in our body. It can also go for very useful electrolyte balance, because the common thing is that we can have the chloride ion with respect to Na plus and K plus and acid base homeostasis, because Cl minus we know is basically the corresponding part what we get from HCl, and we know in our body also the stomach acidity goes down to 2.8 pH is due to that of the formation of HCl. So, chloride can definitely play some acid base homeostasis, that means the environment. So, it is basically therefore a essential electrolyte, that means the anionic part of that electrolyte playing a key role in maintaining the cell homeostasis and transmitting action potential in neurons. So, the transfer of sodium ions from the cell membrane or the neuron, basically neuron will get the signal. So, it is basically the concentration difference. You have the barrier, so cell barrier or the membrane barrier, you have one highly concentrated part and another is low concentrated part. So, you will have the movement. So, that movement will generate a corresponding ionic potential along that particular membrane. So, how this can be transferred? Already we have seen that we can have the proton channel, we can have the sodium ion channel, we can have the potassium ion channel. Similarly, for the first time we are knowing that okay, for the typical inorganic anions, the chloride ion, we can have also the channels, which occur basically in all cell types, in plants, in animals, in bacteria and everywhere. And basically it can permit the transmembrane movement. You have the membrane which is the barrier, and you have to cross that barrier, otherwise you will not have the corresponding differences in the concentrations, because we are talking about that you have highly concentrated one part and you have the low concentration in the other part. So, if you go for that particular movement, and sometime it can so happen that you can have the transfer also from the lower end to the higher end against the corresponding natural process, the osmotic pressure or osmotic flow. So, along their concentration gradient, so rightly we can think of okay, highly concentrated area to low concentration area you can move, but otherwise the reverse can be true if your motor or if your that particular channel and the pump. We know the proton pump, similarly if we can have a typical pump for chloride ions also. So, we can deliver, so we can lift like water from lower to the upper parts, that means low concentration to the high concentration region also. So, CIC channels is basically the chloride ion channels. So, is a huge group of channels, is a huge thing is there. So, we are not going to cover all these things, but only you know what are the CIC channels. Is a basically large family of chloride channels, because depending upon its working thing or depending upon its working potential, you can have many such channels. They are basically giving us a idea that you can have those channels for the transfer of chloride ions in all forms of life. So, basically what we get, earlier we have learnt about the sodium ion and the potassium ion, that you can have the corresponding cationic channel. Similarly, you can have the anionic channel. So, how these anionic channels can be developed, which basically allow the movement of the chloride, that the channel itself tells us that you have the channel, that means you have the passage and you have the plasma membrane of cells. So, plasma is enclosed. So, those cells basically either it is getting chloride ion or it will be losing some amount of chloride ion concentration. So, we call them as you have the transport proteins which are taking care of these ions are faced with an relatively modest challenge of selecting chloride ions, because they are charged. So, if you is controlled by only the potential, the charge and the charge neutralization. Suppose some positively charged species is responsible for trapping the chloride ion, like your sodium ion. So, if you have the sodium ion as well as potassium ion, you do not know, if you are asked to crystallize the sodium chloride as well as the potassium chloride, you should know the corresponding ionic product and the solubility of the individual salts. Similarly, some selectivity, whether we can develop that such that it can discriminate this anion with respect to the other anion, because the phosphate we know the charge is more, size is big, sulphate also your charge is more, size is also big, and the bicarbonate also the charge is less, but the overall charge is a very big one, HCO3 minus is a big one, but the chloride is only a single ionic species, one atom is there, one anion is there, is a Cl minus, and we know the charge relative to that of your corresponding Cl minus ionic radius. And if you have some ionic proteins, because sometimes we can go for the transfer or the transport of those ionic proteins also which are anionic in charge. So, all these things, very small molecule to the big molecule, you must have some selectivity for that particular process. So, what is the difference? We know already that you have the cation channels, say potassium ion channels, and which goes for the selective movement or the gating movement, and which is basically mediated by two things, where we can have the separate structural elements. We know that we can have ionophores, and ionophores we have seen that they are basically crown ethers type of thing. So, those crown ethers type of thing can bind your sodium ion or the potassium ion. So, you can have some structural unit which can take care of the sodium ion or the potassium ion. But what about the CIC chloride ion channels? The selectivity and gating seems to be intimately related. So, if one is affected, the other will definitely be affected. So, the selection of the ion from a pool of other anions is also responsible for the gating process, whether you have a voltage gated process or a ligand gated process for your movement of the chloride ion. So, in the muscle, basically the vertebrate skeletal muscle, CIC Cl minus channels stabilize the resting potential and regulate the electrical excitability. That means, the corresponding electrical pulse, the signal is signal in the form of electrical pulse we can generate from our muscle movement or muscle contraction or muscle relaxation. So, the movement of chloride things can be there even for your muscle contraction and relaxation. So, when we have huge amount of all these things, we know that we go for urination and we remove all these chlorides from our body, the excess amount of these things during our metabolic process. So, we call as a huge subject has been developed also the metabolomics. So, metabolomics will tell you that in kidney is again another vital organ in our body, which can function basically through the transepithelial fluid and electrolyte transport. So, whatever amount of that particular chloride we want to discard from our body, which is not necessary, which is not required, and is basically going from the downstream when we take intake in our food, the food material, it should be discarded from your body. So, it can flow through then again these chloride channels, including one important receptor we call it, we just only know about that, what is GABA A receptor. So, the GABA A receptor is there and can be transported by, these are the abbreviated names also, such that people can understand and know that it is only attached to the chloride transportation, is KCC2 and NKC2. So, N is attached to that transporter only. So, is one type of that particular thing. So, GABA is nothing but gamma aminobutyric acid. So, a type of that transporter is an ionotropic receptor, is not ionophoric or ionophore, is ionotropic, and is a ligand gated ion channel. That means, it is not a voltage gated one. So, the presence of GABA is important. So, when GABA is present, it can take care of the chloride thing, that means, it is gamma aminobutyric acid. So, that butyric acid can show some function or some relationships. So, what is there? So, you have the corresponding ligand. So, endogenous ligand will come, and that endogenous ligand is your GABA, the gamma aminobutyric acid, which is nothing but a major inhibitory neurotransmitter in the central nervous system. So, we are basically correlating these things, very important functions. We know that CNS, the central nervous system for us is important, and the corresponding muscle movement and all this, and we know that the corresponding knowledge for your different availability of the neurotransmitter. So, if neurotransmitter is available, so neurotransmitter giving signals. So, the GABA is basically can inhibit the function of those neurotransmitters, and that can also affect the corresponding passage or the movement of the chloride ions in our body. So, it is basically nothing but, when we know, is a very simplified form or the simplified understanding is that you have the opening up of the thing and the closing of the thing. So, GABA A receptor, A is your superscript. So, GABA A receptor is selectively permeable to Cl minus and to a lesser extent to HCO3 minus. That is why it basically go for the discrimination, that it can selectively choose Cl minus to pass and the bicarbonate anion to block. So, that is the important thing, why we need GABA. So, the structure of this Cl minus selectivity filter, we all know why we use the filter paper, what is called filter, because you have to block something and you have to allow something to pass, that is the corresponding gating function also. What is gate therefore? The gate for your Cl minus ion is the same thing, that you will filter it out, but is a different fashion, not that your filter is there, the ports are there and some ions will pass and some will be blocked. It is not that you cannot have the corresponding filtration where you use the filter paper and chloride will be filtered out and your bicarbonate will be blocked. It is not that, but because the mechanism that filtration is basically depending upon the size of the molecule, size of the neutral species or size of the cation or the size of the anion, if it is allowing some passage like that of your ion selective electrodes. So, it is basically filtering the CLC gas CKS as determined by the cryo-electron microscopy. So, how we determine it? So, one particular substrate or material, people identified it, they have published in some paper, that material has been written in the book. That is why what book we are following the latest informations of all these things. But what instrument they have used basically is a very sophisticated one. So, is a cryo temperature, that means lower liquid nitrogen temperature or liquid helium temperature. So, the cryo-electron microscopy gives us the figure or the picture. So, you have one diagonal arrow, the corresponding arrow for the arrow giving pointing towards the extracellular thing. That means, the movement of the chloride basically, the movement is basically taking place from there, and that movement is basically going from that particular part. This can have the extracellular thing, that means, how you can pass it. So, if you have the, you are blocking this particular part. So, you know about the corresponding protein chain or the peptide chains and the corresponding amino acid residues, because the pendant part of these amino acids, say B16 is the valine or the tyrosine. So, these amino acid residues basically can allow the movement basically. So, the amino acid residues, you see the way it is placed over there. So, is basically the head of these, and is head in the lower side. So, these three basically, when we are talking about these basically, so is valine 166, tyrosine 520 and serine 121. So, serine residues, all we should know, is the hydroxyl function, alcohol type of function, the phenol type of function, and the corresponding valine type of function is there. So, these functions are basically properly oriented within that particular channel. So, not only you should know about the structure, the cartoon diagram of the corresponding polypeptide chain, but also the relative orientations of these three amino acid residues. So, they have shown also in the ball stick representation, and basically that basically showing where you have the arrow. So, that is why the passage is there. So, which is basically indicating by your gray arrows. So, how we therefore have the corresponding CLC, the chloride ion transporter. So, the chloride ion transporter is like this. So, you have the shortened thing of this passage. So, you have the corresponding external chloride ion or the central chloride ion in SEX and S external. So, you have the extracellular side on the top and the cytosolic side on the lower part. So, arrow movement will tell you from left to right you go there. So, you see that you have the passage. So, the passage of these chloride ions, the greens are all your chloride ions. So, it's basically when E is out, that means E is there. So, you look at the E, the positioning of the E, one of the amino acid residues, that once is basically pushing. So, when it is covering and the pushing of the ion to pass through that particular channel. So, one is basically coming out. So, that one is basically coming out, and there is some bump is also there, which is also controlling that particular serine residue. So, that OH, so the channel bump is important, because once it is there, so bump is there. So, only one of the thing, that means one is passing out, but one is still within the above that particular bump. So, what you see now, that you can have the corresponding entry of the proton. So, the proton is then entered, and proton will come to that particular corresponding acid end of the corresponding amino acid residue, and basically it can go for the neutralization of that particular part. So, if you have this particular part, so the protonation of that particular point, the serine residue is still there, will allow the movement of the second chloride ion. So, your second chloride ion will move. So, basically is passage. So, it is a one way movement. So, it is not a catalytic cycle or catalytic loop. The loop is basically for your cycle, where you have moved that thing, then again new chloride ions will come from the top again, and the cycle will be repeated through that particular passage. So, it basically is going for the swinging motion, and we can have the corresponding glutamate gate, and glutamate gate color code, everything is written over there. So, you read it nicely and understand it nicely, what I have explained just now, because these languages are for your knowledge sake only, so that you can preserve it and you read n times until and unless you understand it nicely. So, is basically the gray dashed area. So, the binding site is still there. So, you will basically reaching. So, this is the gray binding site, and the gray binding site is located for your chloride ions to catch over there. So, cystic fibrosis is a transmembrane conduction regulator, which is a complicated one, but you read little bit about that, which is dependent on your chloride channel, and is responsible for transepithelial salt and fluid transport. So, it is very complex one, but is there. But there are people, then even for the newborn babies also, you can have the genetic defect, which can cause a CFTR hypofunctioning, that means lower functioning, not hyper functioning, is a hypofunctioning, lead to cystic fibrosis, is a diseased condition. Sometimes a newborn baby out of 2500 or so, one died. The most common lethal genetic disease. So, how you can correlate a disease, a genetic disease even, to that of your malfunctioning or the hyperfunctioning of a particular regulator, which is dependent on your chloride channel. If we have also the intestinal colonization of the microorganisms, the pathogenics, which are harmful for us, for hyperfunctioning of CFTR. So, these two conditions have been demonstrated over here. One is hypo and another is hyperfunction of all these things, which provokes the secretory diarrhea, is the leading cause of mortality in early childhood. So, another disease can also be identified, which has some relationship with CFTR and which has some relationship with your chloride ions. So, in the next part we will just talk about quickly about the iodine, because the iodine is the most simplest one, and we also know very much about why we take the iodized salt in our food. So, is basically extensively utilized by the marine organisms, because as we have discussed the corresponding source of the chloride concentration in sea water. Similarly, you should also know about the amount what you can have as your source from again the sea water, because iodine always we know compared to your potassium iodide, like your potassium chloride, is a costly salt. So, the algae basically, so brown algal kelp, some part algal kelp species are there, and which are most efficient iodine accumulators. So, they have specified mechanism where the bigger iodide ion it can accumulate, or sometime the free iodine also can be stored, because the reactivity of free iodine, because the I2 as the solid brown black brown solid we know has some different property compared to all other halogens. So, among all living systems is basically it can accumulate, and with an average content of 0.1% of dry weight, representing 30,000 fold accumulation of the element from the sea water. So, it has a huge capacity to trap that particular iodine. So, if we can see or if you can find out that particular algae, you can have a good amount of iodine what you can accumulate from the sea water. So, basically what you find that it is not deposited at the free iodine. So, we are not getting that particular thing, but you have to control, you have to know some geochemistry, you have to know some biogeochemistry, because these two things are coming into the picture together. Once you know that in the salt water, the sea salt water, basically you can have the corresponding iodine or the iodide ion, and the corresponding algae are taking care of all these things, and but it is basically accumulating somewhere. Some sea nodules we can find, and those nodules if we can take out, we can have a plenty of this particular source. So, what we get therefore, that is a required component for your corresponding thyroid hormone. So, how we can correlate why you take the iodized salt, because we have to go for or produce for your corresponding thyroid hormone. What is that thyroid hormone? So, it is basically a phenyl ring which is iodized. So, iodide derivative or iodine derivative of that particular one for the biosynthesis of thyroid hormone basically, and two types of thyroid hormones are required. One is T4, that means 4 positions are iodized, and T3 is 3 positions are iodized based on that hormone thyrionine. So, triiodo species and tetraiodo species, that is why 3 3 prime 5 5 prime and another is 3 3 prime 5 triiodo species with us. So, you see, you try to know about the corresponding structure of these, what is T3 and what is T4 and what is DT and MIT. And looking at the structure immediately you can see that these are some other products, but related to T3 and T4. So, how it can be related to one of the very well known amino acid tyrosine. So, thyroxine hormone is related to the tyrosine residue, because if you see that this particular part, if you protonate it, this is your corresponding tyrosine, the corresponding amino acid. So, the tyrosine amino acid basically, when you can have the further phenyl ring and you can have 2 other positions, that is why the two ortho positions with respect to the O or OH, you have all the iodized positions. So, the DT and MITs are diiodotyrosine residue and monoiodotyrosine residue. So, when you have the corresponding full form of the hormone, you do not have that thing, but if you get this particular part before that iodination, you have the corresponding tyrosine, which is your TYR. So, TYR. So, the tyrosine is your amino acid residue. So, even for that particular process is not the corresponding form, that means another phenyl ring will come, the actual hormone you are getting, but you can go for the corresponding iodination. So, you have the first generated in the thyroid and then circulated throughout the body to regulate many metabolic processes, very important metabolic processes it basically control. So, basal rate of metabolism, how we go for the metabolism, temperature regulation of your body, and expression of numerous other proteins, that means the protein synthesis it is dependent. So, this iodide is transported and concentrated in the thyroid gland. We have the thyroid gland here and here, attaining a level of iodide within the cell 40-fold greater, which is found in your plasma. So, you have to have the corresponding accumulation of these iodide ions. That is why we have the shortage of iodine from our food material. That is why you take through your sodium chloride, iodized salt of the sodium chloride we take, that is your iodized salt, and then it basically going for the bound to the tyrosyl residue of the thyroglobulin. So, thyroid hormone and the globulin is the protein we know, the globin protein like hemoglobin. So, is the globulin protein one form of protein as mono and diiodinated tyrosyl residues in reactions which are catalyzed by thyroid peroxidase, which then serve as the intermediate. So, this monoiodo species and the diiodo species is basically the intermediate. That means, what we have shown just now, that means the two side products are forming always, they are basically required for the final production of your T3 and T4. And the proximal diiodotyrosine residue form an iodothyronil residue product catalyzed by the same thyroid peroxidase responsible for the preceding iodination reaction. So, basically what we get? We have many types of these iodination reactions. So, 7 of them MIT and 6 of them DIT are there, and 1 T4 are formed in the particular that thyroglobulin molecule, and some are therefore can contain T3. So, T3 and T4, and sometime we not only know about the amount of T3 and T4, but also we should know about the corresponding ratio. So, what is that ratio? Because the hydrolysis can take place, and to release the corresponding T3 T4 into the plasma is also required for the proper functioning of your thyroid cells. So, in conclusion, what we have seen now, that your chloride channels may conduct many different ions, can take care, but are named for chloride because its concentration in vivo is much higher than other anions. So, if your concentration is higher, so it will only be transported nicely through proper selectivity. And the deiodination part, the reverse part, the deiodination of T4 to give primarily active T3 proceeds via selenium containing deiodinase is also. Already we have studied selenium. So, that selenium bearing deiodinase enzymes are also available, which can selectively remove the iodine. Not only the iodination, but deiodination is also important for our life. So, chloride channel and the iodine in biology, you read it from the Wikipedia page and the book. Okay. Thank you very much for your kind attention.